So remember, we just created ATP and NADPH in the light-dependent reactions, and now we're going to look at the light-independent reactions, or the Calvin cycle. Notice how we begin with 3-RUBP, ribulose bisphosphate. We add to it carbon dioxide that has to come in through the stomata. We manufacture 6 molecules of 3-phosphoglycerate, and then we have to have an input of 6 ATPs. This is a very energy expensive process. We manufacture 6,1,3-bisphosphoglycerate, and then we have to have an input of 6 NADPH. Now, the end result of that, we're going to make 6 G3P. That's glyceraldehyde 3-phosphate, and that's spelled out below. What is so disappointing, you only get to use one of those, G3P, to make glucose and the other organic molecules. 5 of those have to be used along with an additional 3 ATP just to regenerate RUBP. It's a very, very expensive cycle from the standpoint of energy and raw materials. So in the first part of it, which is called carbon fixation, carbon dioxide combines with RUBP to form a 6-carbon molecule, your 3-phosphoglycerate, and this reaction is catalyzed by the enzyme Rubisco. Rubisco is going to be important to us in a little bit. Then we do carbon reduction, and then finally we regenerate RUBP and form whatever other molecules we're able to. Our sugars, our cellulose, our starches, and we also do what amino acid and fatty acid synthesis is possible. Okay, now we're going to diverge just a little bit and talk about some other things that are related to photosynthesis, for example, sunlight. Sunlight has a wave and a particle nature. Notice that if you look at the electromagnetic spectrum, it goes all the way from very high energy cosmic and gamma rays. There is the part of light that is visible to our eyes, and then down to the lower in energy, radio waves and heat. Now, energy travels in waves. They can be measured, and the distance between two crests of the electromagnetic waves is called the wavelength, and this is the symbol we use for that. And this determines the type of electromagnetic energy we're dealing with. Now, the electromagnetic spectrum is shown above. That's the entire range. But visible light has a wavelength of 380 to 750 nanometers. And these colors can be visualized. And it includes the wavelengths that are used for photosynthesis. When we talk about the particle nature of light, it behaves like particles, and they are called photons. Pigments absorb visible light energy. Different pigments absorb different wavelengths. So there are 3 types of pigments in the chloroplast. Chlorophyll A, that's your key light capturing pigment. Chlorophyll B, which is your accessory pigment. And then we have carotenoids. And they are also accessory pigments. Excuse me. These are the ones that look yellow or orange in fruits and vegetables. The wavelengths that are used for photosynthesis best are the violet blues and red lights. And this was proven by T. Engelmann. And this is called the action spectrum. We know that some of the carotenoids, some of these pigments, actually provide photoprotection, absorbing any excessive light, because remember, that light is also electromagnetic and could cause physical damage to the DNA and to the leaf structure. Okay, so we know the pigments absorb light, and this energy moves them from a stable ground state to an unstable excited state. We're talking about electrons. When the excited electrons fall back to the ground state, they emit heat. And in this particular case, just like most others, this is a wasteful product. That heat is not usable as energy for any other process. There are some pigments that actually emit light, and this is called fluorescence. And we see this especially with things like phytoplankton in the ocean. Now let's talk a little bit more about what a photosystem is. Remember, up here in the light-dependent reactions, we saw photosystem 2, photosystem 1. Excuse me. The photosystem is a reaction center complex plus what we call light harvesting complexes. Now, the reaction center complex is just a bunch of proteins holding a pair of chlorophyll A and a primary electron acceptor. Remember, we have to accept these electrons so they can gain energy from the sun, and then once they're energized, begin to move through this linear electron flow. The light harvesting complex is responsible for the transfer of the energy of photons and chlorophyll to chlorophyll A in the reaction center complex. Primary electron acceptor is in the reaction center, and it accepts excited electrons. Now, let's talk. Whoops. You remember when we were talking about the light-dependent reactions? You notice how the electrons went from photosystem 2 up to P680, down to photosystem 1, up to P700, and then down, and the final acceptor of electrons was NADPH. Well, remember, that's called linear electron flow. And I just want to mention, the reason we call it photosystem 2 and it comes first is because it was found second. The research that had been done actually discovered photosystem 1 first, and then they just figured they could not change the numbers because it would be too confusing in the literature. But I want you to think about something. There is a phenomenon known as cyclic electron flow, where the electrons cycle back from ferredoxin in PS1 via a plastocyanin. Now that sounds like Greek, but all we're saying is that the electrons, rather than doing this whole pathway, they cycle just using photosystem 1. So the end result of that is they produce ATP, but they do not produce NADPH. They don't do that last electron transport chain. They also do not release oxygen. Now this is an interesting phenomenon that we think is very, very ancient in evolution. And we think would evolve before linear electron flow, and it might protect the cells from light-induced damage. But it's just a very interesting phenomenon. So we're going to be talking about some alternative means of carbon fixation that have evolved where it's very hot and dry. Another word for that is arid. And before you say, oh, I really don't care, these plants are pretty or not pretty, we're not talking about the beautiful part. We're talking about the crop plants right now, which are designed to feed the world. So this is serious. Dehydration can be a problem for many plants because on hot, dry days, they're going to close their stomata to try to save water. Well, that makes common sense. But the problem is, then it creates a problem with photosynthesis because you don't have enough carbon dioxide and oxygen builds up. This favors a very poor process called photorespiration. So let's talk about photorespiration. Most plants, and they're called C3, which simply stands for I'm going to be making a 3-carbon compound, initially fix carbon dioxide, just like we talked about in the light-independent reaction, using the enzyme Rubisco. And this forms 3-carbon compounds, specifically 3-phosphoglycerate. Well, Rubisco is a very weird enzyme because it can also utilize oxygen instead of carbon dioxide in the Calvin cycle, and it produces a 2-carbon compound. Now, in photorespiration, it consumes oxygen and organic fuel and releases carbon dioxide, but here's the problem. It does not produce ATP or sugar. We think it's an evolutionary relic when the atmosphere had much more CO2 and not as much O2, and the competition between the two for Rubisco wasn't as big a deal. One of the things that it does, though, it limits the damaging products of the light reactions that can build up if you are not doing the Calvin cycle. But it can be a big problem because on a hot, dry day, it can drain up to 50% of the carbon resources that have already been fixed by the Calvin cycle. So it literally strips out the resources of the plant. C4 plants minimize the cost of photorespiration by incorporating the carbon dioxide into 4-carbon compounds. Interestingly enough, and I want you to go in your textbook and look at figure 10.20. There are 2 types of cells in the leaves. There are bundle sheath cells, and they are arranged literally as sheaths or covers around the veins. And remember, the veins are carrying the water and the food. And then you have mesophyll cells, which are very loosely packed between the sheath and the epidermis of the leaf on either side. Now, as far as sugar production, like glucose in the C4s, the production of 4-carbon precursors in mesophyll is catalyzed by PEP carboxylase, another enzyme. It has a higher affinity for CO2 than Rubisco. So PEP carboxylase is not going to give us that issue with, oh, I'll just combine with oxygen. The 4-carbon compounds are exported to the bundle sheath cells, and they're stored there. And then they release carbon dioxide to run the Calvin cycle. It sounds very complicated, but it works. As the carbon dioxide levels increase here in the world, we know it's going to affect C3 and C4 plants differently. The future evolution or abundance of each of these is really of question and concern. Many of these are crop plants, and especially as suitable land decreases, and we're talking about land that is really suitable for agriculture, and world food demand increases, we've got some challenges ahead of us. There has been some really neat research lately. Excuse me, C4 photosynthesis uses less water and resources than C3. So what scientists have done recently is they took rice, just as an example, which is a C3 plant, and genetically modified it to be a C4. And they have estimated that this alone will increase the yield 30 to 50%. That's pretty incredible when you think about, what the needs are when we're trying to feed the world. So we need to talk about another type of plant called a CAM plant. And the CAM stands for crassulation acid metabolism. And these plants open their stomata at night. They take in CO2 and they use it to make organic acids. And this is stored, these organic acids are stored in vacuoles. During the day, the stomata close because these are things like succulent plants and cacti, and they don't want to lose their water. The CO2 is released from organic acids and goes into the Calvin cycle. So let's just summarize this. The C4 plant structurally separates the initial steps of carbon fixation from the Calvin cycle, but CAM plants, the steps are separated in time. It's a temporal separation. It still occurs in the same cell. And I would go to figure 10.21 and have a look at this. Okay, so what we're going to do first is we're going to review how to do all the genetics problems. We're going to start with just a regular standard Mendelian problem. And so in this case, big T tall is dominant, little t short is recessive. And let's suppose that we have two parents who are heterozygous for the trait, big T, little t. Mom could produce gametes that are big T, little t. Dad could produce gametes that are big T, little t. And when you plug them into a Punnett square, you put mom here or dad, mom here or dad. Doesn't matter, you can put one on one side, one on the other. You're going to do your cross, big T, big T. Remember to always put the capital letter first, like big T and little t. And then big T over here, and little t comes down, and little t comes over, and little t comes down. So what that means is, because tall is dominant, 3 out of 4 of these offspring are going to be tall. That's 75%. 1 out of 4, or 25%, are going to be short. And that's going to give you a ratio of 3 to 1. So even though we have often treated it differently, albinism is another very straightforward Mendelian example, where big A stands for normal pigment, little a stands for the albino condition. So normal pigmentation is dominant. So let's suppose that we have one parent who's homozygous dominant and one parent who is an albino. Well, we know this parent can only produce big A gametes. This parent can only produce little a gametes. And when we put those into a Punnett square, one parent on one side, one parent on the other, we get big A, little a, big A, little a, big A, little a, and big A, little a. Okay? You see how we're doing it? Crossing here to here, here to here. Okay? So that means their children would appear 100% normal, even though they are all carriers of the condition. Now, when you have 100% of anything, then we don't do a ratio. Okay?